Agricultural animals, mainly cows but also chickens, goats, and other livestock, are known to be major reservoirs for E. coli. While not all E. coli is pathogenic, the presence of any E. coli indicates an increased likelihood of pathogenic bacteria being in the area. E. coli is an enteric bacteria, meaning that it lives in the guts of its host. Since agricultural animals are known to be a major reservoir for this bacteria, they are able to pass the E. coli from their guts into the environment through the waste they produce. This waste can then become an exposing factor to human populations if water runoff causes E. coli contaminating waste to come into contact with local water sources, food production areas, or anywhere else humans could possibly come into contact with it. Our case study focused on possible contamination into water sources with the intent that if our hypothesis that there is a correlation between proximity to agriculture and E. coli concentrations in water were accepted, that this can indicate a need for improved regulations on waste management for farms to protect the health of surrounding communities.
In order to test our research question, we collected samples from multiple locations around Massachusetts, including Hadley, Amherst, Braintree, and Springfield. We used sterilized water bottles and submerged them approximately a foot beneath the surface before opening the cap and allowing them to fill with water. We figured that it would be unlikely that E. coli concentrations would be high enough for us to quantify them in the neat or unconcentrated water sample, and so we concentrated our samples by centrifuging them in the lab. And then we plated our samples on McConkey agar. This is a selective and differential medium, which means that it selects for gram-negative bacteria and is able to differentiate between lactose and non-lactose fermenting bacteria. E. coli is a gram-negative lactose fermenting bacteria, and so this would be indicated on our plates by a bright pink colony as opposed to white colonies, which would just be gram-negative non-lactose fermenting bacteria. These samples from Springfield, Hadley, and Amherst were incubated at 37 degrees Celsius for 24 hours before the CFUs or colony-forming units were quantified, and the Braintree samples were not incubated and were kept at room temperature and counted after 48 hours of growth. We were then able to plot our results and test for correlation.
When we collected and recorded our data, we found that the Breckwood Lake location in Springfield was a huge outlier, resulting in 1,979.85 colony-forming units per milliliter and totaled in concentration in 7,062.45, despite being 4.184 miles away from a farm. Because our hypothesis was that increased proximity to farms increased E. coli appearance, we didn't predict for this to happen because Springfield isn't the location that's closest to a farm but had the most colony-forming units. It does suggest that there may be other factors that contribute to E. coli contamination, such as areas being urban or other sources of pollution. In both scatter plots, we graphed bacterial concentration versus distance from farms in kilometers. And on the first graph, it is the comparison with the Breckwood Lake location. And the correlation between proximity and E. coli colonies was very, very weak, with R-squared values being 0.015 and 0.095 for total concentration in blue and CFUs per milliliter in red, respectively. However, when we took out the Springfield location in the second scatter plot, the correlation became a much stronger negative linear correlation, with R-squared values being 0.698 and 0.558 for total concentration and CFUs per milliliter, respectively.
Although our hypothesis was not supported by our collected data, some valuable conclusions can still be made about water contamination and quality through the results of our study. Even though Breckwood was not the closest location to a farm, it had the highest level of E. coli concentration. This outlier brings light to the fact that water contamination can happen for a multitude of reasons, not just due to agricultural pollution. At the same time, it is important to recognize the stronger correlation between proximity to farm and E. coli concentration when the Breckwood point is disregarded. Although correlation does not equal causation and more data must be collected to make a definitive statement, it is likely that proximity to farms has an effect on E. coli concentration. This research is important because there is a lack of open information about the testing done on bodies of water in Mass, and it was unknown how living in or near a farming community can affect water quality. With the results collected, it seems that bodies of water near farms should be regulated and frequently tested. But generally speaking, any body of water near any potential source of pollution should be tested frequently for the safety of all communities. Thank you for watching.